Thanks, Danny. The title of the idea was colorimetric path tagging of filaments using DNA-based metafluorophores. And to give a slight analogy to it, the basic idea operates a little bit like the photograph on the title slide. You essentially take filaments running through certain stations and paint them. And look at the color they accumulate at the end as a way of doing the path. So I'll try to keep this bit short just to avoid redundancy. The requirements Danny's already talked about briefly. When I looked at those, it seemed to very strongly incentivize two things. One was trying to pack as much information density as possible into each individual tag, and also to come up with some way to be able to do a single shot readout rather than a sequential readout, or as close to single shot as you could get. And for me, the only sensible option to continue considering as a concept for this would be DNA, simply because you have, with 4 base pairs, the ability to build some fairly large amount of information into each sort of individual piece of thing that the filament carries. And then I also initially toyed with the idea of trying to sequence, but figured that it would need to be more single shot than that. So I sort of floated around the literature for quite a while thinking about how you might do this sort of thing. And I guess there's 2 papers that were big inspiration for my idea. One was a paper in 2017 that basically looked at trying to make color-based tags out of DNA origami using short sort of staple strands as handles to grab onto short pieces of DNA with fluorophores attached to them. And I sort of had a passing curiosity in DNA origami for quite a few years now, mostly due to a colleague of mine, Lawrence Lee at UNSW, who works on this. So I've sort of been following the literature and saw this paper and thought, oh, this is a really clever way to get high density, very quick readout of a high density of information in some sort of way. And so the basic idea they have here is they have a DNA origami tile with a large number of sort of just short single-stranded DNA pieces hanging out of the tile. You bind on fluorophores. You can direct particular colors to particular parts of the tile by setting the sequence they're attached to. And so you can build up a tile that basically accumulates some amount of red, some amount of blue, and some amount of green. And much like the pixels in a television screen, if you make the right mixture of red, blue, and green, you can make pretty much any color that you imagine. And so the nice thing that they showed in this paper was basically the ability to color DNA origami tiles a little bit like a rainbow under a fluorescent microscope, entirely off the basis of how many of pixels of red, green, blue, and they also added in purple to mix it up a little bit. And you can see that they get a very large spectrum for information density into what is essentially a very, very small package, which is in some ways a nice way to use DNA, both to increase your information density, but also in this particular case to get single shot readout, because simply by looking at what color that particular tile is, you can work out with quite a lot of accuracy how much red, how much blue, how much green, and how much purple, and then turn that back into some piece of information that you want. The second piece of inspiration that came up for this was I saw a talk that Shelley Wickham, who used to be in William Shee's group at Harvard, was giving when she moved back to Sydney. And she was talking about these DNA barrel structures. And you can see in the micrographs down the bottom here that you get these really nice, long, ring-shaped DNA barrels with a dimension that's kind of quite nicely around the diameter that you would have for a microtubule, possibly if you push it far enough, even an actin filament. And the nice thing about these is that you can put sort of little extending staple strands of DNA, both on the inside and hanging on the outside. And you can separate them by about 9 to 10 nanometers or so and have a very high density of these on both the outside and the inside. So the idea I had was basically to try and take some of the barrels that you have here, put them on the outside of a microtubule, essentially a little bit like a t-shirt, have it make its way through the maze, and the t-shirt starts out white in the sense that it's got no colored tags attached to it at all. And then it moves through a network that features a set of, I guess what you would call color stations, where you have DNA tagged to fluorophores, and those bind to staple sites on the outside of the barrel on the filament, and basically attach a color to it. So when the filament goes through the green station, it picks up a green fluorophore. When it goes through the red station, it picks up a red one. And depending on what path it takes, it could, at the end of that, have 3 times green plus 4 times blue plus 3 times red. And that would give it a particular color that you could then, with a single shot measurement, read what the color is and then work backwards to what the path was that was taken. So just drilling into this in slightly more detail, the barrel diameter, as I just mentioned, fits kind of quite nicely to the scale, particularly of the microtubule. One of the nice aspects of the idea is that you can get a reasonable number of tags on the outside with barrels that only partially cover the microtubule. So you essentially would still have around about 80% of the microtubule exposed on the outside, available for example, kinesin to grab on and pass it along the system. And then some small number of areas where there's little patches where you just can't bind to it because the DNA sort of gets in the way. And so you could imagine having not just a nice sort of smooth cotton t-shirt, but something that's a little bit like a hairy Velcro t-shirt, where you have a set of staples, DNA staples strands sticking out on the inside. You could bind those to short single strand DNA pieces connected to some sort of filament binding protein. So something like alpha-tubulin or phalloidin for an actin filament. And this has kind of been demonstrated. So there was a nice paper from Douglas et al. In 2012 that basically bound proteins on the inside of a DNA origami and then locked it shut with sort of a lock and key mechanism. And their focus was on how to take this thing that was locked shut and unlock it. But in this case, you could just take the first part of that. You could have one of these barrels that would be an open object and basically have it fold around the outside and then zipper shut to hold it in place on the filament while it travels through the system. And it might even be possible to use such a lock and key mechanism so that when you get to the exit, you can trigger a key that unlocks the t-shirt and makes it fall off the filament so that you could direct it to some other place to read. And then the outside would basically be a very large number of these short staple strands devoted to binding on fluorophores so that you could carry your color information. And the one big difference that we would have to the Warstein paper, in their one, all of the staple strands that were on the outside of the tile had exactly the same sequence. What you would do here is have them with different sequences so that they would be very specific to picking up a particular color at a particular station in the system. And you could use that as part of the way of disentangling the path information later on in the setup. So it wouldn't be a good idea without some sort of hand-drawn sketches. So this is some of the drawings from early parts of the concept. So to draw it up, it would kind of look a little bit like this. You could imagine having sort of typical filament length is sort of 5 microns or so. You might imagine to get a reasonable number of bits in the system that you might carry something like 15 of these DNA barrels along the length of the system, and that would still leave you with about 80% of the surface free for binding to the motors, so that the system still works as a sort of motile filament concept. And one thing that you could do is build a little bit of redundancy into this system. So of the 15 t-shirts, for example, you could devote 3 of them to just carrying a whole pile of fluorophores, so that essentially your filament and every single one of them carries a standard candle that allows you to calibrate the optical measurement further down the system. And then the other 12 carry your tagged information for what's going on. And that would in principle give you something like about 320 bits of information. And if you were willing to give up the 3 for the standard candle, then you would get about 960 distinct bits, which sort of kind of pretty well meets what's required in this. And you could probably pretty quickly build in some other things to get further bits. So for example, in the Warstein paper, they talked about using photostability as a piece of information. So what you do is you take 2 dyes that have very similar color, but very different photostability, and you look at how they decay as a function of time. And you could do the same thing here. You could basically use 2 colors with slightly different photostability to build a little bit more information, carrying capacity into one of these metafluorophores. So then the next thing after that is basically how do you paint and how do you read the paint? And the painting probably, in my view, would be best done by trying to build little paint stations in your network. And one way to do that would be to have a front plane on the structure that's essentially a microfluidic, and multi-level microfluidic structures are reasonably well developed now. That could be prefabricated onto essentially a nanoporous polycarbonate substrate, which basically acts as a dialysis membrane. And the idea behind the dialysis membrane is you want your fluorescent tags to be able to get into the system, but you don't want them to come in such a flood that they basically diffuse everywhere and every station ends up painting brown rather than one station painting red and one station painting green. So essentially they're there as a sort of a fluidic impedance management for the paint, so that it sort of comes in as a trickle and diffuses out of the system, and it's there in a sufficient density local to your station, but not such a high density that it diffuses throughout the system and paints everything up. And this is one of these structures that you could in principle bond together. You could probably make the microfluidic front pane on the polycarbonate first and then sandwich it on top of the typical sort of nanochannel structures that currently get used for these microtubule assays. And on top of that, you still have to worry about tags diffusing through the system. So one thing I was thinking about here was that you could have some sort of chemical countermeasure for fluorescent tags that have managed to stray away from their paint station. So if you think back to the color run concept, you spray paint around, imagine you have 2 stations that are close together and you've got a very windy day. And one of the issues that you will have with these kind of network computing devices is you basically need to be able to feel sort of flow nutrients through the system. You need to be able to supply ATP and other things. There might be a drift and the paint will drift downstream. So basically I wanted to work out a way to deactivate paint that gets to where it's not supposed to be. So the idea for this is basically that in a lot of these assays, you have a surface that's coated in protein, essentially to stop your filaments from sticking to the bottom surface. And one possibility would be to use those surface proteins to basically hold on to a little anti-handle, which is the red structure that you see in here. It's essentially just a little short DNA strand that is complementary to this green strand here, which basically is something I call a deactivation strand. So you pre-prime your system with these, and you have a background of them floating around as a replacement mechanism. And what you do is you arrange for the other end of this deactivation tag to be complementary to the tail of your paint strand. And assuming that this part of the strand is complementary to part of this green, you should be able to use a strand displacement behavior to basically have the paint tear off this deactivation strand, completely deactivate itself by properly binding onto that strand. It gets flushed out of the system, so you still have the paint, but the paint can't bind to your filament. And then this short anti-handle here is basically designed just to pick up a new deactivation tag and reset the trap so that it keeps working, capturing paint as you go on. So in addition to dialysis membrane, you should be able to deactivate anything, you should be able to stop the paint from getting away really quickly, and if it does, you should be able to capture it and deactivate it. And then the last part of the idea is basically how do you do single shot readout. And one of the issues that in principle you have here is that if you have a top structure on, you can no longer really use a microscope. And for practical applications, you probably don't want to use a microscope. And there's been quite a bit of work in the last few years to look at developing CMOS backplanes that are capable of super resolution microscopy. So essentially you operate in a near field sort of structure. And for a device like this, if you wanted a full CMOS backplane everywhere, would probably be rather expensive. But what you probably need is only pixels local to where you want to be able to read what the output is. And those could be quite small and localized on the base of your chip. So there's already been a little bit of work on this, looking at on-chip detection of DNA binding to surfaces colored with quantum dot fluorophores, and they can do detection down to 20 fluorophores per square micron for 50 by 50 pixel size. And with the number of fluorophores that you have on the outside of one of these tags, you should be able to detect. And that's 2009 standards for that kind of microscopy. There's some predictions that basically suggest that 1 fluorophore per square micron is probably reasonable to do in the near future. So this might be one viable way to measure this. And one initial concern that I had pretty straight up was, well, now your fluorophore is kind of wrapped around the outside of your filament. So some of the light's going to be shining out the top. How do you measure that? One of the nice aspects in particular of microtubules is that they tend to rotate as the filament passes along through the system. And so if your CMOS pixel for detection is longer than that rotation distance, which is typically about 10 microns or so, you should be able to essentially capture light from all the parts of the barrel on the outside of the filament and still be able to do a full readout of that structure in that way, providing the pixels long enough. So that's pretty much most of the details of the idea. So I think I might stop there.